Why saving microbes might be the most crucial conservation endeavor ever. A groundbreaking study has been released, detailing how researchers collaborated to craft the initial comprehensive strategy for safeguarding microbial life. Led by Professor Jack Gilbert, president of Applied Microbiology International, this initiative marks a significant milestone in global conservation efforts. The research is featured in the article Safeguarding Microbial Biodiversity, Microbial Conservation Specialist Group, MCSG, within the Species Survival Commission of the International Union for Conservation of Nature, IUCN, published in Sustainable Microbiology, an AMI journal, initiating a global endeavor through the IUCN. In July 2025, the IUCN officially established the MCSG within its Species Survival Commission, co-chaired by Professor Gilbert and Raquel Pixoto, CAUS/ISME. The group emerged from a workshop in May, which brought together conservation experts and microbiologists to explore the application of traditional conservation goals in a world dominated by microbial processes. This marks the first global coalition dedicated to protecting microbial biodiversity, the invisible 99% of life, ensuring that microbes are recognized as vital to the planet's ecological, climate, and health systems, Professor Gilbert stated. He continued, I believe this shifts conservation from protecting individual species to preserving the intricate networks of invisible life that make visible life possible, marking a paradigm shift towards planetary health. It also provides us with a comprehensive view of microbial tools that can support conservation actions, enabling us to utilize microbiology to address the world's most pressing challenges. The significance of microbial biodiversity for the planet. Microbes play a pivotal role in soil fertility, carbon cycling, marine productivity, and the health of plants and animals. Despite their critical importance, they often remain absent from conservation policies. Professor Gilbert emphasized that neglecting microbial diversity undermines climate resilience, food security, and ecosystem restoration efforts. He explained, the MCSG addresses this gap by integrating microbiology directly into the IUCN's conservation framework, utilizing red list criteria, ecosystem assessments, and restoration programs to ensure microbes are considered in policy, not just in scientific research. Building a global network for microbial conservation guidance. Over the past 2 years, the founding members have assembled an international community of microbiologists, ecologists, legal experts, and indigenous knowledge holders from over 30 countries. Together, they have developed the first microbial conservation roadmap, outlining 5 key components of the IUCN species conservation cycle. Assessment. Developing metrics for microbial communities and biobanks compatible with the Red List. Planning. Establishing ethical and economic frameworks for microbial interventions. Action. Piloting restoration projects utilizing microbial solutions, such as coral probiotics, soil carbon microbiomes, and pathogen-resistant wildlife. Networking. Connecting scientists, culture collections, and indigenous custodians worldwide. Communication and policy. Launching public and policy campaigns, including the initiative Invisible but Indispensable. Initial efforts are supported by funding from the Gordon and Betty Moore Foundation, along with in-kind support from AMI and ISME. This phase focuses on mapping microbial hotspots, creating conservation indices, and integrating existing microbial biobanks into a coordinated global archive. Overcoming scientific and ethical challenges. Professor Gilbert acknowledged that establishing a conservation framework for microscopic life presents complex scientific and conceptual barriers. These challenges include defining what constitutes a microbial species within Red List criteria, integrating genomic and ecological data into systems originally designed for plants and animals, addressing the belief that microbes are too resilient or complex to require protection. Microbial conservation must navigate an immense unseen diversity and highly dynamic community structures that defy classical species concepts. Taxonomic instability, the lack of long-term baselines, and the ethical handling of microbial samples, including indigenous or human-associated microbiota, all necessitate new definitions of loss, restoration, and rights of microbes, he stated. He added, however, A significant milestone was achieved when the IUCN approved the MCSG as a formal specialist group, officially extending global conservation to microbes for the first time. Plans for the next phase of conservation endeavors. The group's upcoming goals include developing the first microbial red list framework by 2027, creating global maps of microbial hotspots across soil, marine, and host-associated systems, testing conservation strategies such as microbial bioremediation, coral probiotics, and soil carbon restoration, ensuring microbial indicators are integrated alongside plants and animals in IUCN and UN biodiversity targets by 2030. What is required for long-term progress? Sustained investment is crucial to expand global microbial monitoring networks. Future progress also relies on integrating microbes into national biodiversity and climate strategies, including 30 by 30 foot and One Health policies. Another key priority is fostering public microbial literacy, recognizing microbes as the foundation of ecosystem and human health. The roadmap also emphasizes the importance of digital twin and AI tools to predict how microbial communities will respond to environmental changes. The paper was published today, November 20th, in Sustainable Microbiology,